Thank you for the kind introduction. So I'm Joanna from Genome Institute of Singapore. And today I'm going to talk about the SG10K SV project, whereby we generated a catalogue of structural variations in 10,000 Singaporeans. So what are structural variations? So structural variations are genomic rearrangements of more than 50 base pairs, and they can be classified into different classes such as your deletions, the duplications, the insertions, the inversions, and the translocations. Because of the sizes of the structural variations, they tend to impact a large fraction of the human genome, and therefore they are also significant contributors to diseases and traits. So currently at present, there are many large consortiums around the world that profile the structural variations across individuals of different populations around the world. However, these databases are mainly Eurocentric focus. So Asia, with with a population size of which accounts for 60% of the world populations, they are underrepresented in these databases, and therefore very little is known about the genetic diversities in Asians. And therefore, to bridge the knowledge gap, the National Precision Medicine Program, which aims to sequence the genome of 10,000 healthy Singaporeans, we started this program, also known as the SG10K Health. So the SG10K Health dataset contains individuals of the Malay, Chinese, and Indian ancestries in Singapore. And one of the key features of this SG10K Health dataset is that it contains Malay genomes that have never been included in any of the previous large-scale SV studies. So because this SG10K Health dataset also comprises of datasets from different sub-cohorts that are sequenced at different sequencing depth and also the different library construction methods, which can influence SV detection accuracy. So we split our dataset into a discovery dataset and also two validation datasets. For the SG10K SV project, we started by calling only the three most common structural variation types: the deletions, the insertions, and the duplications. These three different structural variation types, they have very different genetic properties, and therefore it is very difficult to identify all three of them using a single analytic tool, unlike single nucleotide polymorphisms. So in order to identify the three different SV classes, we use different tools such as we use Mantas femur graph typer for deletions and insertions. For the mobile element insertions, we use a very specialized tool known as MELT to call them. And lastly, for duplications, we used an in-house tool called SurfIndel tool, whereby it has a higher sensitivity in calling duplications compared to Mantas femur graph typer pipeline. And so this is the pipeline that we use to identify the structural variations in our SG10K dataset. So using the SG10K SV pipeline, we managed to identify a total of 152,000 structural variations in approximately 5,487 individuals, which are in the discovery dataset. When we compare these structural variations with Nomad SV, we found that 75% of these variants are novel, and this reflects the potential for new discoveries by analyzing underrepresented Asian genomes. So apart from identifying novel structural variations, we are also interested to identify Asian-specific SVs. So we calculate the fixation index. We use the fixation index method to identify Asian-specific SVs, and we found that 50% of the structural variations are Asian-specifics. So the two notable examples that we see will be a deletion in intron 2 of BIM gene, which is associated with TKI resistance, and we also found a rare known deletion spanning the HBA1 and HBA2 gene that is associated with alpha thalassemia. So We are also interested to know the percentage of the structural variations that cover the individual genomes. So on average, each individual within the SG10K health, they harbor around structural variations covering 0.41% of the genome. In contrast, SNPs and short indels, they cover only 0.17% of the genome. So this shows that structural variations, in fact, they affect a much larger fraction of the genome compared to SNPs. And therefore, it is very important to characterize these structural variations in the human genome. So we also look at the number of variants detected in each genome for each individual, and we found that majority of the variations are deletions, followed by duplications and insertions. When we look at the allele count distribution of the SVs, similar to all the previous consortium studies, we found that majority of these structural variations are actually rare variants with less than 1% allele frequencies. We also look at the length distribution of the SVs that we detect in our dataset. We are able to identify the three very distinctive peaks, the ALU, the line 1, as well as the SVA peaks, and they actually highlight the pervasive contributions of repeat elements in scalping the human variations. Majority of the SVs that we see in our datasets, they are very small. They are small, less than 10 kilobase, which is expected of short read sequencing data. However, we also do identify deletions and duplications longer than 10 kilobase pairs in our dataset. Next, we also look at how structural variations can impact regulatory elements as well as the gene bodies. So we first look at the enrichment and depletions of different structural variation class in the ENCODE CCRE regions, and we found that common deletions, they are significantly depleted at putative enhancer and insulators regions, which is consistent with a model of negative selection and acting on alterations affecting gene expression. We also found that common insertions, they are depleted 11-folds at coding regions. And similar to enhancers, we found that rare and ultra-rare structural variations, so rare structural variations are variations less than 1% allele frequency, and ultra-rare is less than 0.01% allele frequencies. They show weaker depletion patterns in exons of all types. And when we look at the intronic regions, we see that most of the structural variants do not have any deviations from the background, except we see some elevations, modest elevations in rare and ultra-rare insertions. We also want to assess the functional consequences of structural variations on coding genes. So SVs deleting gene regions may cause loss of functions events, and similarly for duplications, they may lead to copy gain, which will affect your gene dosages. So when we look at our data set, we find that 3% of the structural variations, they actually have direct impact on the protein coding integrity. Around 4,000 of them are likely to cause a loss of functions, but this loss of function structural variations, they tend to occur at very low allele frequencies. When we look at duplications, we found around 1,000 duplications that may cause copy gain in one or more of the protein coding genes. So this will actually cause gene dosages changes. And then we also found that copy number gain events, they tend to be larger compared to loss of functions events. So we also look at how structural variations can affect clinically actionable genes. So we annotate our SVs with the ACMG version 3.1 gene list. And we found that out of all the structural variations that we detect in our data set, 35 of them will actually affect protein coding sequence integrity of the 21 of these ACMG genes. So we found 2 deletions in this TRDN gene in Chinese individuals only. So here's an example of the SAM plot. The SAM plot showing an example of the deletion, the TRDN gene. So the first 2 rows shows individuals with the deletion, and the last 2 rows shows individual without the deletions. And yeah, so we found only Chinese individuals having these deletions in the TRDN genes. And then we also found another deletion affecting 48 individuals in the DSG2 gene in our data set. So both this TRDN and DSG2 genes, they are associated with severe cardiac dysfunction. Okay, so we also look at the principal component analysis of our SV's genotypes, and then we found that we are able to recapitulate the PCA that is generated using SNP-only dataset, and this shows that there are genetic variations between the 3 ethnic groups in Singapore, and therefore we are interested to identify ethnic-specific structural variations within our dataset. So we calculated we used the fixation index method to identify ethnic-specific structural variations, and we found 144 of them that shows ethnic-specific differences. So some examples of known SVs will be we saw a deletion in ACOT1 gene that's involved in fatty acid metabolism. So this variant is found at a lower allele frequency in Indians. And then we also found several novel Asian-specific SVs, such as a deletion in the AHNAK2, which has a high prevalence in Indians, and also a deletion in the TNNT3 gene, which has a higher allele frequencies in Malays compared to the other ethnic groups. Yeah, lastly, we also try to identify structural variations that are in linkage disequilibrium with GWAS lead SNPs. So we hypothesized that some of the GWAS lead SNPs that we see in all the GWAS studies that are associated with certain traits, they may not be the causative variants, and the causative variant might actually be a SV that is in strong linkage disequilibrium with these SNPs. So by performing this analysis, we found 75 of these SVs that are in strong linkage disequilibrium with the GWAS lead SNPs. One example would be a deletion in the GPP3 gene that's in strong linkage disequilibrium with this GWAS lead SNPs that's involved in subclinical atherosclerosis in Chinese individuals. So all in all, we show that the SG10K-SV dataset contains 75% of the variants are novel in our dataset, and we are able to identify Asian-specific variations. We demonstrate the use of SG10K-SV to identify ethnic-specific structural variations, and we also show how you can use the SG10K-SV catalog to find map Asian GWAS catalogs. So all in all, the SG10K-SV resource and its analysis shown here augment global knowledge of structural variations across human populations, and they will direct participate to reduce current ancestry bias in genetic discoveries. Lastly, this project would not be possible without the people involved in the SG10K projects, and we also like to thank all the cohort owners, TTSH, Gusto, Helios, Prism, NUS, and SID, as well as the National Supercomputing Center for supporting and for providing resources and supporting us for the analysis of this SG10K project. Lastly, we also like to thank our funders, the IFPP and the National Precision Medicine Phase 2 funding. Thank you.
Thank you, Joanna. So we are open to the questions. I guess this is a very useful resource and valuable resource for the analyzing our Asian genome structure variation. Yes, please.
Hi, so fabulous talk. I have a few questions, but yeah, just some technical details. So which reference genome did you use?
GRCH38.
Have you considered like T2T because you mentioned repeat regions and all those?
Yes, but maybe in the next phase.
Makes sense. So for the long SVs, how do you, because I think it's short read, right?
Yes.
How do you confirm or validate the ultra long structure variations?
How we confirm? At present, we didn't do any validation yet, but we will check them against IGV.
Okay, I see. Last question then. It's very interesting to see like there's structure variation in strong LD with the GWAS SNPs. It can like inform many interesting mechanisms. But like, how do you know which one is causative? Like, did you do some statistical analysis on like whether it's the SV or the SNP?
So at present, we can only tell if the SV is in linkage disequilibrium with the SNP. But in order to know if it's causative, we will need to further validate them. You definitely need to do some functional studies to check if it's the SV that is the causative. And you also need to do some large-scale GWAS studies using SVs to correlate with your SNPs as a validation.
I see. So these ones might not be available through like UK Biobank and other big data set that you can try to confirm these. Are these SVs just specific to this population and not they do not exist in other big data sets such as UK Biobank, because you know you can use those to try to find map and confirm your hypothesis.
I think for now, because we only focus on the Asian data set, as in when we do the LD computation, we only do it against Asian catalogs. So some of our SVs can also be found in NOMAD. So I think you can do the same analysis in like using the UK Biobank dataset, but they may not have a lot of Asian SVs like in their database.
Yeah. Okay. Thank you.
Thank you.
Yeah, I have just one little question. Is this database already available for for the public, or still have not published?
Okay, so once our paper get accepted, this
Okay,
resource will be put on the NPM National Precision Medicine website, Chorus browser, and you can download the entire data set down.
Okay, thank you very much.
Any other? Actually, I have a one quick question. So you compared your data to the NOMAD dataset, but I'm not sure if all Asian genome data are available in NOMAD dataset, or for example, Japanese or Chinese datasets existing.
I think in NOMAD, so far we saw East Asians, we saw South Asians. I think there are some Koreans and Japanese genomes within NOMAD.
Because a comparison within Asia region is also interesting.
Yeah.
Okay, thank you.